Okay, well, welcome to the Classics of Immunology Journal Club. And before we get started, I just want to remind you to like it on the YouTube site, subscribe to the channel, and click on the bell and get notified of more of these. And then also scroll down from on the YouTube site and go on to my website, and you'll find, if you click on the writing tab, you'll find all the classic papers of immunology, including the one I'm going to discuss today. And the paper I'm going to discuss today is really a landmark paper in immunology, I think. The authors are Kevin Tracy and Bruce Beutler. And Bruce Beutler's portrait is behind me here. He's one of the famous gliterates that I've chosen to paint their portrait. And then there's 8 more people. And then at the end of the series of authors is Tom Schreier's, and then finally Anthony Cerami from the Rockefeller University. And the title of this paper is Shock and Tissue Injury Induced by Recombinant Human Cachectin. It was published in Science in 1986. And the background on this paper is that this was a collaboration between Tony Cerami's group and Bruce Beutler at the Rockefeller, people across the way at Cornell Medical School. Tom Schreier was the chairman of surgery there, and Kevin Tracy was a neurosurgical trainee. I don't know whether he was a resident or a fellow or what, but he was a young guy, and he was in the lab, and he was the one that was responsible for I assume, for most of the in vivo experiments that I'm going to tell you about. Meanwhile, Bruce was by this time, and Tony was a biochemist, and they had already purified cachectin at this stage of the game. And I reviewed their paper where they showed basically that cachectin and whatever tumor necrosis factor were the same thing. Cachectin is a funny word. It relates back to the fact that Tony got involved in this whole process because when he was a young blade, he got a fellowship and went to Africa, and he was struck by all of the patients that had chronic ongoing infections because they were so wasted. They were just skeletal. They looked like they'd been in Auschwitz, essentially. Question was, why was that? And that's how Tony got involved in trying to understand that from a biochemistry standpoint, and ultimately found that when patients had chronic infections, and or if he injected experimental animals with lipopolysaccharide from the cell wall of bacteria, that they would get the same syndrome. So that's sort of the background on that whole thing. It's interesting that Kevin Tracy's the first author. He did these in vivo experiments, which is really the crux of this particular paper. But Bruce Beutler was the driving force as far as I'm concerned about this whole thing. And Bruce is, even though he was listed second author on the paper, he was footnoted to the fact that he was the corresponding author. And so if you wanted to find out more about this paper, you were supposed to write to Bruce. And by this time, Bruce had left Tony Cerami's lab and had taken up his first assistant professorship at the University of Texas Southwestern in Dallas, which is where Bruce trained as an intern and a resident, his clinical training. So there's one other author on the paper that's important, and that's James Merriweather, who was a scientist at Chiron, which is a biotech company on the West Coast. And they were the source of they used recombinant tumor necrosis factor cachectin for these experiments, and basically it came from Chiron. The introduction of this paper it had been known since like 1850 that, well, even before that, if you got septic with bacteria, and in particular the bacteria that are very common in the colon, if those bugs got into your blood, you basically were marked for death, and there were really no therapies that could prevent it, and you went downhill very rapidly. Blood pressure dropped, you go into shock, you have multi-organ system disease, and you die. Now, the active component of that was figured out in the 1850s or in the 1880s and 90s by one of Robert Koch's students at the Koch Institute in Berlin. and it was found to be a cell wall component of these bacteria that had the characteristics of lipopolysaccharide, abbreviated LPS. And so if you purified LPS, and there's a lot of work that had been gone on ever since in the entire 20th century to find out what it was about this LPS that was so toxic and so forth. But it was known by then, if you had pure LPS and you injected it in experimental animals intravenously, that they would undergo extremely high fevers, shaking chills, and go into shock with a falling blood pressure, and then end up with multi-system organ failure, as I've said. They would die, and at autopsy, they would have what was labeled shock lung. Basically, they had disseminated interstitial pneumonia in this shock lung. And they had acute tubular necrosis in their kidneys, and their whole GI tract was infarcted and hemorrhagic. Basically, that's what happens when the blood pressure goes down. Now, the fellow, and I don't have his name, I'll have to get that for you, but the fellow who first identified LPS called it has an alternative name, which is endotoxin. By then, it was known that there are some bacteria and bugs that would sort of release into the culture medium or whatever surrounding them, what they called an exotoxin. So it was an excreted external product that was toxic. Now, tetanus toxoid, tetanus toxin is one of them. Diphtheria toxin is another. Well, LPS was thought to be released from inside the cell by the early bacteriologists, and so that was why it was called an endotoxin. It came from the inside of the bacteria, and so for years thereafter, for I think most of the 20th century until this paper, it was felt that LPS was causing the symptoms of shock, death, and so forth and so on itself. It was the toxin. However, this paper dispenses that whole idea because it is an endotoxin, but the endotoxin is not coming from inside the bacteria. It's coming from inside the infected host. That's a critical insight in terms of how we get sick and how we might get better if we can get effective therapies for these kinds of things. So that's the background. At the time, well, no, there's more background in the intro. By the time that Bruce and Tony and the others were doing these experiments, there were two different strains of an inbred mouse called C3H. One was C3H/HEJ, and that was resistant to LPS. You couldn't kill them by injecting LPS into them. But another strain, C3H/HEN, was sensitive, and you could kill them by injecting LPS into them. That turns out to be very important later on. But it meant that, and they traced them, they used those mice and they traced the whole deal that it was the hematopoietic cell in the mice that was responsible for the toxicity, and that the macrophage was really a critical player in this whole process. Bruce and Tony had already shown that cachectin was a macrophage-derived protein, and when they found out that it interacted with their target cells by means of specific receptors, they started calling it a hormone, which I think is also very critical in terms of how one thinks of different molecules in the body and how they might work. And they had already shown that cachectin would decrease fat uptake into fat cells because they inhibited the activity of lipoprotein lipase, which was an enzyme on the surface of fat cells that would digest triglycerides into individual fatty acids so that they could be taken inside. And at the same time, and Bruce had shown this, that the same cachectin that he was purifying would cause lysis of tumor cells, some strains of tumor cells that were in cultures in vitro. And in vivo, if you injected animals with purified or semi-purified cachectin, the tumors would undergo hemorrhagic necrosis. That's how the name tumor necrosis factor was coined by Lloyd Old back in the 70s. And they found out that there was a huge amount of protein coming out of these macrophages when you stimulated them with LPS, as much as 1% to 2% of the total protein coming out of those macrophages. The question then became, is it the LPS that's causing this toxicity when you purified that and injected it in animals, or was it something that the LPS was doing to the host that was causing this whole thing to happen? And they thought that the likelihood was that it was the cachectin or the TNF, and it was not the LPS itself. So in the materials and methods of this paper, they got recombinant TNF from Chiron out in California. And they had done, at Chiron, they had engineered the recombinant product so that it would be highly expressed in yeast. So you could get huge amounts of recombinant protein out of the yeast. And they had engineered the whole system so that it was, so that the TNF or the cachectin was 30% of the secreted protein coming out of the yeasts in culture. That was a good deal. So Bruce had worked out all the biochemical techniques to purify cachectin, and they used the same biochemical techniques that I went to in some of the previous papers to purify this recombinant TNF molecule. And it was critical that when they measured their purified product, that it would not be contaminated by lipopolysaccharide. That's one of the reasons they made it out of yeast instead of E. coli in cultures. And they could show that it had minuscule, very, very small amounts of LPS in their preparations through specific LPS assays. And so what they did was they would take this purified protein that they ran through their biochemical processes, and now we should go into screen sharing, is the end result of their biochemical purification processes. And this is a SDS polyacrylamide gel that separates proteins on the basis of their molecular size. And they had a single band on an SDS page, and that was their purified product. So that's what the first thing they did was and this is what I think Kevin Tracy had already established in rats. It was a system to be able to test their product. And this is survival among Sprague-Dawley rats 12 hours after treatment with cachectin in various doses. And they started from 0 and they went up to 3.6 milligrams per kilo of body weight of the rats. And then this is at 12 hours after the treatment. These are the number of rats in each group, and this is the percentage of survival at 12 hours. And you can see that when they were at low doses, 0.2 milligrams, all the animals survived. When they went up to 0.6 milligrams, only 1/3 of them survived. And the mean survival time was close to 7 hours. They went up even higher. It was still about 1/3 survived, but now they died around 5. Mean survival time was 5 hours. And they went up to 3.6 milligrams. Nobody survived. It was even less time of the ones that did survival time was 4 hours. And this is the last one is an important one because they had a monoclonal antibody reactive to their product at this time, and they treated them 1 hour with their monoclonal antibody and then injected them with the highest dose of the cachectin. And you can see that 5 of 5 survived, 100% survival, and they were fine. So that was what they were looking for. That was their specificity in this thing. And going back to the first page, this figure 3 here is the gross appearance of the GI tract in an animal that died 4 hours after cachectin infused at 1.8 milligrams per kilo. And you can see that what's happened here, this is just the intestine taken out of the animal. And this is what hemorrhagic necrosis of the intestine looks like. The whole intestinal wall is basically hemorrhaged. That becomes very, very important when we're thinking about this whole shock syndrome. And I'll get into that later. So that was just one example of their whole process. And then I want to show you is they did some histology then. And this is in this column here on the left is lung, and in the middle is intestine, and over here on the right is kidney. Down at the bottom is our control animals that were not injected with cachectin, and here at the top are the ones that were injected with cachectin. And so if you look at the lung, you see all these air spaces here. They look just fine and so forth. And this is just a blood vessel here. And you can see now in this lung, there's the air spaces are basically obliterated by small round cells, and they were both polys as well as red cells. There was hemorrhage into the lung. And that was the shock lung, basically. Over here is the intestine. And you see there's nice villi coming into the lumen of the intestine and so forth. Here's the mucosa or the muscular layer, everything, it looks good. And in here, you see in between, first of all, the villi are all gone. They don't even have that in there. It should be up here. And in the submucosa, it's just filled up with inflammatory infiltrates, essentially. Over here in the kidney, these are the glomeruli. And the normal glomeruli are here. And this is normal kidney with tubules and cells lining the tubules. And you can see what's happened here is that the cells are basically that should line the tubules are basically gone. This is what acute tubular necrosis looks like, essentially. So I think that's all I want to really show you. There's other things that they have in terms of graphs and tables in the paper of the toxicity, but this really brings it home. Yeah. And so I'll stop the screen share at this point. So if you have purified recombinant TNF or cachectin and you inject it into these poor rats, they get really sick and then they die. They start in their discussion and they say, because of the fact that they've already discovered that cachectin TNF interacts with specific receptors and mostly on all the cells of the body, they said then you should expect to have multiple effects of this hormone on the body. Exactly what's happened here. And they say that there's, in the history of medicine and the history of science, there's no single endogenous molecule that would really do this until this particular paper. Now, they had already shown in other basic science experiments that there was a cascade of what we now recognize as pro-inflammatory cytokines. And the TNF or cachectin was really at the top of the cascade because it was found to cause macrophages to make interleukin-1. And interleukin-1 would make T cells and NK cells, or not NK cells, T cells primarily, and make interleukin-2 and also interleukin-6. And so TNF, IL-1, and IL-6 are now what is considered the pro-inflammatory cytokines. And when they found this out, they already had monoclonal antibodies reactive to TNF, cachectin, and they wondered whether or not they could block sepsis. And so they did a lot of experiments to see if they could do that. And certain biotech companies got involved in the whole process, and they tested in the clinic whether their antibody would block septic shock in hospitalized patients. It's a clinical trial now, and they really couldn't show that happened. That they couldn't improve the survival curves of people that were treated with their antibody versus not. And they basically put it on the shelf. And it wasn't until a couple of groups, Mark Feldman being one of them in England, who was studying rheumatoid arthritis, wondered whether or not the chronic administration of TNF and the blockage then therefore would help in a chronic infection kind of situation. And that's where the anti-TNF worked. And it went on them. They started using it for Crohn's disease and ulcerative colitis and psoriasis and a lot of these chronic, weird inflammatory syndromes. If you would block TNF, you could have a significantly beneficial effect on these diseases. So that was good. And that all came subsequently to this paper, but this paper was the kingpin of the whole deal. The other thing is, though, in terms of, I showed you what happened in the lung when you just injected TNF into these rats. So that's what the lung looks like in COVID, where you have the symptoms of COVID are basically systemic inflammation. The whole body becomes red, hot, swollen, and sore, and it's due to the host reaction to the virus. The so-called prodromal period of the virus, you get exposed, you get infected, and then there is this asymptomatic period where you don't have any symptoms, but the virus is replicating and replicating and replicating. Well, that tells you that the virus doesn't cause any symptoms. It's only when the adaptive immune system kicks in and starts making all these pro-inflammatory cytokines that you start having problems. And so if you're going to treat COVID with anti-inflammatory drugs, you need to do a shotgun approach with, I think, with steroids, glucocorticoids, dexamethasone, which is what our wonderful President Trump got, is one of the drugs of his therapy. The other thing is they need to give at the same time. So glucocorticoids will block production of these cytokines, but the cytokines that are already there is not going to do anything for. So you need to block the cytokines that are already there, and you need to use anti-TNF, anti-IL-1, anti-IL-6, in a cocktail. That's Kendall's solution to the severe COVID infection. The other thing that I think is very important is that other kinds of this so-called septic shock syndrome have been now figured out to be the release of these pro-inflammatory cytokines. For example, the high doses of interleukin-2 that have been used in cancer therapy by Steven Rosenberg's group at the NCI cause exactly the same kind of syndrome. And in particular, the GI tract, you get a severe colitis. And after I showed you that picture of what the intestines of the rats look like after just a simple injection of TNF, it's perfectly understandable, because what high doses of IL-2 do is that they kick off a whole cascade, primarily coming out of the NK cells that have IL-15 receptors and memory T cells that have IL-15 receptors. IL-2 will bind to those cells at very high doses, high concentrations, and stimulate a cascade of pro-inflammatory cytokines to take place. So with LPS, it activates primarily initially macrophages. IL-2 hits NK cells and memory T cells. And then more recently in this whole mix has been so-called CAR T cells, CAR, which stand for chimeric antigen receptor T cells, where they put a receptor into T cells that has an antibody molecule on the outside that will bind to a tumor cell, and on the inside of the T cell they have some of the chains that activate the whole T cell activation process. And when somebody has a lot of tumor burden, and then you inject lots and lots of CAR T cells, you get a huge clash, and you get release of all kinds of cytokines, the triumvirate of TNF, IL-1, and IL-6 particularly. And one of the first toxicities that they saw when they gave these CAR T cells was the whole septic shock syndrome, which they called cytokine storm. We have a new word for it. Every time each generation of doctors have to give a name to these things, because unbeknownst to them, it's already been discovered back in 1986. the checkpoint inhibitors that were used. The colitis was a major problem early on. They couldn't understand why these poor people were having just liters and liters of bloody diarrhea. So that's the story. I'll stop there. Don't forget to like the video if you like it, and subscribe to the channel. Click on the bell. Check out my website and my social media sites. We talk about a lot of different kinds of things there. So thanks so much for your attention.